Hi, I'm Dr. David Green, founder and CEO of R3 Stem Cell, the global leader in regenerative therapies. Today, we're discussing natural killer cell therapy. I'm here with one of our top physicians at our Tijuana, Mexico clinic, Dr. Oscar. How are you doing today?
I'm doing good. How are you?
Excellent, thank you. So, Dr. Oscar, can you tell me about the types of treatments with NK cells that we do here at the Tijuana clinic?
Sure. So basically what we do is put healthy NK cells from a donor through IV or in situ in the tumor per se. So we apply basing on the age, the type of cancer, and also their weight. That's very important aspects to make sure to to have a good dosage of NK cells to make them work properly.
So once you get the donor blood,
Yep.
How do you work with that blood in the lab to kind of separate out the natural killer cells and then expand them, but B, make sure that the blood is safe?
Okay, so all of our blood is tested previously to see they don't have any kind of disease, any kind of immune response, any kind of something that's going to affect our patients. And once we separate the blood, we separate the red cells from the white cells. So what we want right here is the white cells, because that's where the NKs are located.
Right.
Then we separate all the kinds of white cells because we have lymphocytes, we have T cells, B cells, and any other kinds of stuff. But what we want are the NKs.
How do you separate the NK cells from the other types of lymphocytes?
Okay, so we have some markers that help us to get to the NKs only. These are some specific markers that are called CD16. So we put some antigens, let's say, in our cells to make sure what cells are NK cells and what cells are not NK cells.
Okay, and then with the CD markers, you can see which ones are the NK cells. And then is it a filtration method? How is it that those are separated from the others?
Yeah, we have different filtration methods. We have centrifuges that help us by their molecular weight, and also these antigens that I was telling you about.
Yes.
And that's how we separate the NK cells from other type of cells. Once we have only the NK cells, our biologists right here cultivate these cells and expand them, because we don't want only, let's say, 1 million of NK cells. We want 100 million, 200 million to do a good therapy to make a change in the patient.